probably know you need to eat protein. But what is it? Many foods contain protein, but the best sources are beef, poultry, fish, eggs, dairy products, nuts, seeds, and legumes like black beans and lentils. Protein builds up, maintains, and replaces the tissues in your body. Your muscles, your organs, and your immune system are made up mostly of protein. Your body uses the protein you eat to make lots of specialized protein molecules that have specific jobs. For instance, your body uses protein to make hemoglobin, the part of red blood cells that carries oxygen to every part of your body. Other proteins are used to build cardiac muscle. What's that? Your heart? In fact, whether you are running or just hanging out, protein is doing important work like moving your legs, moving your lungs, and protecting you from disease.
Dear children, today we will learn about the structure of proteins. Proteins are probably the most important class of biochemical molecules and are the basis for the major structural components of animal and human tissue. Proteins are natural polymer molecules consisting of amino acid units. The number of amino acids in proteins may range from 2 to several thousand.
On completion of this topic, learners will be able to list few sources of protein, write and discuss different structure of proteins, discuss the formation of peptide bond, discuss alpha and beta helix in proteins, compare between primary, secondary, tertiary and quaternary structure of proteins, discuss effects of physical and chemical effects on proteins, discuss effects of heat on proteins.
Protein ਵਿੱਚ molecular mass, amino acids ਦੇ complex biopolymers ਅਤੇ organic compound ਹੁੰਦੇ ਹਨ ਜੋ ਸਾਡੇ ਜੀਵਨ ਲਈ ਬਹੁਤ ਜ਼ਰੂਰੀ ਹਨ। ਇਹ 20 ਤਰ੍ਹਾਂ ਦੇ amino acids ਨਾਲ ਬਣੇ ਹੁੰਦੇ ਹਨ ਜਿਨ੍ਹਾਂ ਨੂੰ ਬਾਰ-ਬਾਰ ਪ੍ਰੋਟੀਨ ਦੀ ਬਣਤਰ ਵਿੱਚ ਵੇਖਿਆ ਜਾ ਸਕਦਾ ਹੈ। ਜਦੋਂ ਪ੍ਰੋਟੀਨ ਹਾਈਡਰੋਲਾਈਜ਼ਡ ਹੁੰਦੇ ਹਨ ਤਾਂ ਇਹ amino acids ਆਜ਼ਾਦ ਹੋ ਜਾਂਦੇ ਹਨ। ਪ੍ਰੋਟੀਨ amino acids ਦੇ polymers ਹਨ। All proteins contain the elements carbon, hydrogen, oxygen, nitrogen and sulfur. Some of these may also contain phosphorus, iodine and traces of metals like iron, copper, zinc and manganese. The name protein is derived from the Greek word proteus, meaning of prime importance. Amino acids are the building blocks of proteins. Amino acids contain two functional groups. They are amino and carboxyl groups. The amino group is basic and the carboxyl group is acidic in nature. Amino acids are zwitter ions. An amino acid has both a basic amine group and an acidic carboxylic acid group. There is an internal transfer of a hydrogen ion from the COOH group to the NH2 group to leave an ion with both a negative charge and a positive charge. This is called a zwitter ion. This is the form that amino acids exist in even in the solid state. If you dissolve the amino acid in water, a simple solution also contains this ion. Adding an alkali to an amino acid solution. If you increase the pH of a solution of an amino acid by adding hydroxide ions, the hydrogen ion is removed from the NH3 positive group. We can now show that amino acid now exists as a negative ion using electrophoresis, adding an acid to an amino acid solution. If we decrease the pH by adding an acid to a solution of an amino acid, the COO negative part of the zwitter ion picks up a hydrogen ion. This time, during electrophoresis, the amino acid will move towards the cathode, shifting the pH from one extreme to the other. Suppose we start with the iron we have just produced under acidic conditions and slowly add alkali to it. That iron contains two acidic hydrogens, the one in the COOH group and the one in the NH3 positive group. The more acidic of these is the one in the COOH group, and so that is removed first, and you will get back to the sweeter iron. So, when we have added just the right amount of alkali, the amino acid no longer has a net positive or negative charge. That means that it wouldn't move towards either the cathode or anode during electrophoresis. The pH at which this lack of movement during electrophoresis happens is known as the isoelectric point of the amino acid. This pH varies from amino acid to amino acid. General structure. The carboxyl group and amino group are attached to the same carbon atom, hence amino acids are termed as alpha amino acids. Let us now have a look at the peptide bond formation.
Peptides are compounds formed by condensation of two or more or different alpha amino acids. As mentioned earlier, amino acids are the building blocks for proteins. All amino acids contain an amino or NH2 group and a carboxyl acid or COOH group. There are 20 different amino acids commonly found in proteins and often 300 or more amino acids per protein molecule. Each amino acid differs in terms of its R group. The R group of an amino acid is the remainder of the molecule, that is, the portion other than the amino group, the acid group, and the central carbon. Each different amino acid has a unique R group, and the unique chemical properties of an amino acid depend on that of its R group. To form polypeptides and proteins, amino acids are joined together by peptide bonds, in which the amino or NH2 of one amino acid bonds to the carboxyl acid or COOH group of another amino acid. Polypeptides are formed by when a large group of alpha amino acids are joined by peptide bonds.
Structure of proteins. Let us now recall what we have studied so far. Peptides are compounds formed by the condensation of two or more or different alpha amino acids. The condensation occurs between amino acids with the elimination of water. In this case, the carboxylic group of one of the amino acid and the amino group of another amino acid gets condensed with the elimination of water molecule. The resulting CONH linkage is called peptide linkage or peptide bond. Primary structure of proteins. Proteins are biopolymers containing a large number of amino acids joined to each other by peptide linkages having three-dimensional structures. The structure of proteins is a complex one which is divided into four parts. They are primary, secondary, tertiary and quaternary structures. Primary structure. Proteins may have one or more polypeptide chains. Each polypeptide in a protein has amino acids linked with each other in a specific sequence. This sequence of amino acids is said to be the primary structure of that protein. So, the sequence in which the amino acids are linked in one or more polypeptide chains of a protein is called the primary structure of proteins.
The linear sequence of amino acids in a polypeptide chain represents the primary structure. Each polypeptide chain has a free amino group at one end and free carboxyl group at the other end. This structure is responsible for the function of a protein. This structure is composed of various amino acids held together by peptide bonds. Proteins may have one or more polypeptide chains. Each polypeptide in a protein has amino acids linked with each other in a specific sequence, and it is this sequence of amino acids that is said to be the primary structure of that protein.
The primary structure is usually determined by its successive hydrolysis with enzymes or mineral acids. The amino acid sequence of a protein determines its function and is critical to its biological activity. Frederick Sanger determined the primary structure of protein for the first time in insulin in 1953. The importance of the primary structure lies in the fact that even a change in one amino acid can drastically change the properties of the entire protein. It also creates a different protein. For example, a normal hemoglobin has 574 amino acids units, and changing just one amino acid in the sequence results in a defective hemoglobin found in the patients of sickle cell anemia. In the patients suffering from sickle cell anemia, the defective hemoglobin in red blood cells precipitate, causing the cells to sickle and sometimes even burst, leading ultimately to death. Let us now have a look at the animation to know more about normal hemoglobin and sickle cell hemoglobin.
ਆਮ ਤੌਰ ਤੇ ਹੈਰੀਡਿਟੀ ਡਿਸੀਜ਼ ਸਿਕਲ ਸੈੱਲ ਅਨੀਮੀਆ ਦੇ ਕਾਰਨ ਹੀ ਰੈੱਡ ਬਲੱਡ ਸੈੱਲਸ ਅਤੇ ਹੀਮੋਗਲੋਬਿਨ ਦੀ ਸ਼ੇਪ ਵਿੱਚ ਐਬਨੋਰਮੈਲਿਟੀਜ਼ ਆਉਂਦੀਆਂ ਹਨ। ਸੂਖਮਦਰਸ਼ੀ ਹੇਠ ਇਹ ਇਨਫੈਕਟਿਡ ਰੈੱਡ ਬਲੱਡ ਸੈੱਲਸ ਦੀ ਸ਼ੇਪ ਕ੍ਰਿਸੈਂਟ ਵਾਂਗ ਹੋ ਜਾਂਦੀ ਹੈ ਅਤੇ ਇਹ ਹੋਰ ਮੁਰਝਾਏ ਹੋਏ ਲੱਗਦੇ ਹਨ। ਇਸੇ ਕਾਰਨ ਇਹਨਾਂ ਨੂੰ ਇਹ ਨਾਂ ਦਿੱਤਾ ਜਾਂਦਾ ਹੈ। ਇਸ ਬਿਮਾਰੀ ਨਾਲ ਇਨਫੈਕਟਿਡ ਸੈੱਲ ਦੀ ਸ਼ੇਪ ਰੈੱਡ ਬਲੱਡ ਸੈੱਲਸ ਵਾਂਗ ਬਾਈਕੌਨਕੇਵ ਸ਼ੇਪ ਨਹੀਂ ਹੁੰਦੀ। ਇਸ ਦੇ ਸਰਫੇਸ ਏਰੀਆ ਵਿੱਚ ਕਮੀ ਹੋ ਜਾਣ ਕਾਰਨ ਆਕਸੀਜਨ ਦੀ ਟਰਾਂਸਪੋਰਟੇਸ਼ਨ ਟਿਸ਼ੂਜ਼ ਅਤੇ ਆਰਗਨਸ ਵਿੱਚ ਐਫੀਸ਼ੀਐਂਟਲੀ ਨਹੀਂ ਹੋ ਸਕਦੀ। ਆਓ ਨਾਰਮਲ ਬਲੱਡ ਫਲੋ ਤੇ ਇੱਕ ਨਜ਼ਰ ਮਾਰੀਏ। RBC ਸਰੀਰ ਦੇ ਡਿਫਰੈਂਟ ਪਾਰਟਸ ਵਿੱਚ ਆਕਸੀਜਨ ਪਹੁੰਚਾਉਣ ਦਾ ਕਾਰਜ ਕਰਦਾ ਹੈ। ਸਿਕਲ ਸੈੱਲ ਅਨੀਮੀਆ ਦੇ ਕੇਸ ਵਿੱਚ ਜਦੋਂ ਕਿ RBC ਵਿੱਚ ਲੋ ਆਕਸੀਜਨ ਕਨਸੈਂਟ੍ਰੇਸ਼ਨ ਹੋ ਜਾਂਦੀ ਹੈ ਤਾਂ ਉਹ ਸਿਕਲ ਦੀ ਸ਼ੇਪ ਦਾ ਹੋ ਜਾਂਦਾ ਹੈ ਅਤੇ RBC ਦੇ ਸਟਿਕੀ ਹੋਣ ਕਾਰਨ ਕੈਪਿਲਰੀ ਦਾ ਬਲੌਕੇਜ ਹੋ ਜਾਂਦਾ ਹੈ। ਇਹ RBC ਦੇ ਲਾਈਫ ਸਪੈਨ ਨੂੰ ਘਟਾ ਕੇ 120 ਦਿਨਾਂ ਤੋਂ 60 ਤੋਂ 70 ਦਿਨ ਦਾ ਕਰ ਦਿੰਦਾ ਹੈ। ਸਿਕਲ ਸੈੱਲ ਅਨੀਮੀਆ ਦੇ ਹਾਨੀਕਾਰਕ ਪ੍ਰਭਾਵ ਵਜੋਂ ਹੀ ਗੋਲ ਸਟੋਨਸ ਬਣਦੇ ਹਨ। ਇਲਾਜ ਨਾ ਮਿਲਣ ਤੇ ਦਰਦ ਹੁੰਦੀ ਹੈ, ਇੱਥੋਂ ਤੱਕ ਕਿ ਆਰਗਨ ਵੀ ਡੈਮੇਜ ਹੋ ਸਕਦਾ ਹੈ।
Let us now have a look at the genetic basis of sickle cell anemia, which is as shown on the screen. To learn better about it, let us view an animation again.
Let us now have a look at the genetic basis of sickle cell anemia. Hemoglobin is found in red blood cells. The hemoglobin molecule is a tetramer consisting of 4 polypeptide chains known as globins, which are usually 2 alpha chains that are each 141 amino acids long, 2 beta chains that are each 146 amino acids long. Attached to each chain is an iron-containing molecule known as heme. Oxygen is transported in combination with the iron molecule of the heme group. Let us now have a look at the protein's foldings, which consists of amino acid sequences. In case of a normal amino acid sequence, the 6th amino acid is glutamic acid. Due to mutation in DNA, sequence of CTT and GAA becomes CAT and GUA. As the amino acid sequence is altered, valine is the new amino acid instead of glutamic acid. resulting in altered structure of protein and abnormal hemoglobin having sticky ends, which can carry less of oxygen. The result of fibrillization of RBC, and there is change in conformation of RBC to sickle shape.
Secondary structure of proteins. Let us now learn about the secondary structure of proteins. This structure explains the folding of a polypeptide chain. Two different types of secondary structures are observed. Alpha helix. The alpha helix model was postulated by Linus Pauling in 1951. Resonance takes place between different peptides linkages which depends upon nature of the peptide bond. Due to the partial double bond character of the CN bond in peptide linkage, the amide part, that is CONH, is planar and rigid, that is no free rotation about this bond is possible.
The free rotation of a peptide chain can only occur around the bonds joining the nearly planar amide groups to the carbons. The angles shown as phi and xi in the figure are known as Ramachandran angles after the name of the Indian biophysicist G.N.A. Ramachandran. Note that C double bond O and NH groups of the peptide bond are trans to each other. Hydrogen bonds between NH and C double bond O groups of peptide bonds give stability to the structure. Thus, a structure having maximum hydrogen bonds shall be formed.
Beta pleated sheets. These are formed either by separate polypeptide chains or by a single polypeptide chain folding back onto itself. Two neighboring chains are held by intermolecular hydrogen bonds. Beta sheets may be arranged either in parallel or anti-parallel. N-terminal are aligned head to head, that is, on the same side in the parallel beta configuration, and are aligned head to tail. All the chains are interbound and forms a flat sheet-like structure and hence called beta pleated sheet structure. The beta sheet is parallel in keratin, the protein present in hair, and anti-parallel in silk fibroin.
आओ वूल विच प्रेजेंट अल्फा हेलिक्स ते चात पाइए। ਇਹ ਇੱਕ ਰਾਈਟ ਹੈਂਡਡ ਕੋਇਲ ਹੈ ਜਿਸ ਵਿੱਚ ਇਨਫਰਾ ਚੇਨ ਹਾਈਡਰੋਜਨ ਬੌਂਡਿੰਗ ਹੈ ਅਤੇ ਨਾਲ ਹੀ ਇੱਕ ਮਾਈਕਰੋ ਲੈਵਲ ਤੇ ਐਲਫਾ ਹੈਲਿਕਸ ਸਟਰਕਚਰ ਵਿੱਚ ਐਟਮਸ ਨੂੰ ਵੇਖੀਏ ਜਿਨ੍ਹਾਂ ਨੂੰ ਡਿਫਰੈਂਟ ਕਲਰ ਨਾਲ ਦਿਖਾਇਆ ਗਿਆ ਹੈ। ਆਓ ਸਭ ਤੋਂ ਪਹਿਲਾਂ ਹੈਲਿਕਸ ਦੇ ਇੱਕ ਟਰਨ ਨੂੰ ਵੇਖਦੇ ਹਾਂ। ਇਹ 5.4 ਐਂਗਸਟ੍ਰੋਮ ਦੇ ਡਿਸਟੈਂਸ ਤੇ 3.6 ਰੈਸੀਡਿਊਲਸ ਪਰ ਟਰਨ ਵੀ ਦਿਖਾ ਰਿਹਾ ਹੈ। Spiders produce a variety of silks, and the cloning of genes for silk fibroins reveals a clear link between protein sequence and structure property relationships. The fibroins produced in the spider's major ampullate gland, which forms the dragline and web frame, contain multiple repeats of motifs that include an 8 to 10 residue long polyalanine block and a 24 to 35 residue long glycine rich block. These proteins are present in form of plates and are arranged in an anti-parallel way.
Let us recall what we have learned while studying the secondary structure of proteins. The secondary structure gives the manner in which the polypeptide chains are arranged. So, it gives the shape or conformation of the protein molecule. This arises from the plane geometry of the peptide bond and the hydrogen bond between the carboxyl group and the NH group of different peptide bonds. Pauling and Corey investigated the structures of many proteins with the help of X-ray patterns. It was observed that there are two common types of structures. One, alpha helix structure, and second, beta pleated sheet structure. Let us now have a look at the tertiary and quaternary structure of proteins. Tertiary structure of proteins. The three-dimensional structure of a protein is known as tertiary structure of a protein. This is a compact structure that is further folding of the secondary structure. It coils and folds in such a way that the hydrophobic side chains are held interior and the hydrophilic groups are held outside. This arrangement gives stability to the molecule. The tertiary structure is maintained by hydrogen bonds, disulfide bonds, ionic bonds, and hydrophobic interactions. This structure brings distant amino acid side chains nearer. Major molecular shapes are found. They are fibrous and globular proteins. The fibrous proteins, for example, silk collagen and alpha keratins, have large helical content and have rod-like rigid shape and are insoluble in water. We will have a look at the structure of collagen triple helix in the animation. In globular proteins, for example, hemoglobin, the polypeptide chains consist partly of helical sections which are folded about the random cuts to give it a spherical shape. The primary, secondary, tertiary and quaternary levels of hemoglobin structure are shown in detail in the animation. Let us have a look at it now.
When proline, an oddly shaped amino acid, occurs in the polypeptide chain, a kink in the alpha helix develops. Kinks can also be caused by repulsive forces between adjacent charged R groups. These kinks create a three-dimensional chain arrangement that is the tertiary structure. This three-dimensional shape is also held together by weak hydrogen bonds and also by much stronger disulfide bonds between two amino acids of cysteine, that is covalent, disulfide bridges, that is linkages. Other strong linkages such as ionic bonds can be formed between regions of the same polypeptide, creating the tertiary structure. And additional weaker interactions contribute to the overall three-dimensional shape of the protein. These strong covalent bonds hold the protein in its specific 3D shape. The 3D shape creates pockets or holes in the surface of the protein which are very important in enzyme function.
Let us have a quick recap of what we have learned so far. Tertiary structure arises due to folding, coiling and bending of polypeptide chains producing three-dimensional structures. This structure gives the overall shape of proteins. In other words, the tertiary structure of a protein gives the overall folding of the polypeptide chains, that is, further folding of the secondary structure. Two major molecular shapes found are fibrous and globular. The fibrous proteins such as silk collagen and alpha keratin have large helical content and have rod-like rigid shape and insoluble in water. We saw the helical structure of collagen triple helix.
आओ ਹੁਣ ਕੋਲੇਜਨ ਟ੍ਰਿਪਲ ਹੈਲਿਕਸ ਤੇ ਇੱਕ ਝਾਤ ਪਾਈਏ। ਕੋਲੇਜਨ ਲੌਂਗ ਫਾਈਬਰਸ ਸਟਰਕਚਰਲ ਪ੍ਰੋਟੀਨ ਵਿੱਚੋਂ ਇੱਕ ਹੈ ਜਿਸ ਦੇ ਫੰਕਸ਼ਨ ਗਲੋਬੂਲਰ ਪ੍ਰੋਟੀਨਸ ਜਿਵੇਂ ਕਿ ਐਂਜ਼ਾਈਮਸ ਨਾਲੋਂ ਬਿਲਕੁਲ ਵੱਖਰੇ ਹੁੰਦੇ ਹਨ। ਕੋਲੇਜਨ ਦੇ ਟਫ ਬੰਡਲਸ ਨੂੰ ਕੋਲੇਜਨ ਫਾਈਬਰਸ ਕਿਹਾ ਜਾਂਦਾ ਹੈ ਜੋ ਕਿ ਇੱਕ ਐਕਸਟਰਾ ਸੈਲੂਲਰ ਮੈਟ੍ਰਿਕਸ ਦਾ ਮੇਜਰ ਕੰਪੋਨੈਂਟ ਹੈ ਜੋ ਜ਼ਿਆਦਾਤਰ ਟਿਸ਼ੂਜ਼ ਨੂੰ ਸਪੋਰਟ ਕਰਦਾ ਹੈ ਅਤੇ ਉਹਨਾਂ ਨੂੰ ਬਾਹਰ ਤੋਂ ਸੈਲ ਸਟਰਕਚਰ ਦਿੰਦਾ ਹੈ। ਕੋਲੇਜਨ ਕਈ ਸੈੱਲਾਂ ਦੇ ਅੰਦਰ ਵੀ ਵੇਖਿਆ ਜਾਂਦਾ ਹੈ। ਕੋਲੇਜਨ ਵਿੱਚ ਗ੍ਰੇਟ ਟੈਂਸਾਈਲ ਸਟਰੈਂਥ ਹੁੰਦੀ ਹੈ ਅਤੇ ਇਹ ਫੇਸ਼ੀਆ, ਕਾਰਟਿਲੇਜ, ਲਿਗਾਮੈਂਟਸ, ਟੈਂਡਨਸ, ਬੋਨ ਅਤੇ ਸਕਿਨ ਦਾ ਮੇਨ ਕੰਪੋਨੈਂਟ ਹੁੰਦਾ ਹੈ। ਮੁਲਾਇਮ ਕੈਰਾਟਿਨ ਦੇ ਨਾਲ ਹੀ ਇਹ ਸਕਿਨ ਦੀ ਸਟਰੈਂਥ ਅਤੇ ਇਲਾਸਟਿਸਿਟੀ ਵਾਸਤੇ ਵੀ ਜ਼ਿੰਮੇਵਾਰ ਹੁੰਦਾ ਹੈ ਅਤੇ ਇਸ ਦੇ ਡੀਗ੍ਰੇਡੇਸ਼ਨ ਨਾਲ ਰਿੰਕਲ ਸਪੈਨ ਲੱਗ ਜਾਂਦੀਆਂ ਹਨ। ਇਹ ਬਲੱਡ ਵੈਸਲਸ ਨੂੰ ਸਟਰੈਂਥ ਦਿੰਦਾ ਹੈ ਅਤੇ ਟਿਸ਼ੂ ਡਿਵੈਲਪਮੈਂਟ ਵਿੱਚ ਮਹੱਤਵਪੂਰਨ ਭੂਮਿਕਾ ਨਿਭਾਉਂਦਾ ਹੈ। ਇਹ ਕੋਰਨੀਆ ਅਤੇ ਅੱਖਾਂ ਦੇ ਲੈਂਸ ਵਿੱਚ ਕ੍ਰਿਸਟਲਾਈਨ ਫਾਰਮ ਵਿੱਚ ਮੌਜੂਦ ਹੁੰਦਾ ਹੈ। ਇਸ ਨੂੰ ਕਾਸਮੈਟਿਕ ਸਰਜਰੀ ਅਤੇ ਬਰਨਸ ਸਰਜਰੀ ਵਿੱਚ ਵੀ ਵਰਤਿਆ ਜਾਂਦਾ ਹੈ।
Quaternary structure of proteins. Children, let us have a look at the last structure of proteins. This last level of organization is simply taking two or more three-dimensional and sticking them together to form a larger protein. Many enzymes in transport proteins are made of two or more parts. Hemoglobin, an oxygen-carrying protein in red blood cells which is made of 4 parts. Let us have a look at the animation to know more about proteins.
Many proteins exist as a single polypeptide chain, but there are some proteins which exist as assemblies of 2 or more different polypeptide chains called subunits. These subunits may be identical or different. These are held together by non-covalent forces such as hydrogen bonds, electrostatic attraction, and van der Waals interactions. The best known example of protein processing quaternary structure is hemoglobin, which transports oxygen from the lungs to the cells and carbon dioxide from the cells to the lungs through the bloodstream. It is an aggregate of 4 polypeptide chains or subunits. 2 identical alpha chains containing 141 amino acid residues and 2 identical beta chains each containing 146 amino acid residues. These 4 subunits lie more or less at the vertices of a tetrahedron. Each polypeptide chain carries a hemigroup at its end. To sum up, let us now have a look at the primary, secondary, tertiary, and quaternary structures of hemoglobin.
Effect of heat on protein. The most energetically stable state of a protein is called its native state. In this state, the protein possesses maximum hydrogen bonds. In other words, this state corresponds to the proteins found in a biological system with a definite configuration and biological activity. Proteins get denatured when they are heated. It brings physical and chemical changes in the protein. Heating increases the thermal vibration of the molecule, disrupting hydrogen bonding. The denatured protein will lose its biological activity. Denaturation does not change the primary structure. During denaturation, the protein molecule uncalls from an ordered and specified conformation into a more random conformation, and protein precipitates from solution which may or may not be reversible.
Effect of heat on protein is an irreversible denaturation process. Heat can be used to disrupt hydrogen bonds and non-polar hydrophobic interactions. This occurs because heat increases the kinetic energy and causes the molecules to vibrate so rapidly and violently that the bonds are disrupted. The proteins in eggs denature and coagulate during cooking. Other foods are cooked to denature the proteins to make it easier for enzymes to digest them. Medical supplies and instruments are sterilized by heating and denature proteins in bacteria and thus destroy the bacteria. When the temperature is cooled, the protein does not return back to its original shape, which also means that it loses its function.
Effect of physical and chemical treatments on protein. Protein ਦੀ denaturation ਵਿੱਚ secondary ਅਤੇ tertiary structure ਦੋਹਾਂ ਵਿੱਚ disruption ਅਤੇ possible destruction ਹੁੰਦਾ ਹੈ। ਜਦੋਂ ਕਿ denaturation reactions ਇੰਨੇ strong ਨਹੀਂ ਹੁੰਦੇ ਕਿ ਉਹ peptide bonds ਨੂੰ ਤੋੜ ਸਕਣ। Denaturation process ਦੇ ਬਾਅਦ ਵੀ primary structure ਵਿੱਚ amino acids ਦੇ sequence ਪਹਿਲੇ ਵਰਗੇ ਹੀ ਰਹਿੰਦੇ ਹਨ। Denaturation प्रोटीन ਦੇ अल्फा हेलिक्स ਅਤੇ बीटा शीट्स ਨੂੰ डिस्टर्ब करके एक रैंडम शेप ਵਿੱਚ अनकॉइल कर ਦਿੰਦਾ है। Denaturation. Denaturation occurs because the bonding interactions responsible for the secondary structure and tertiary structure are disrupted. In tertiary structure, there are 4 types of bonding interactions between side chains, including hydrogen bonding, salt bridges, disulfide bonds and non-polar hydrophobic interactions which may be disrupted. Therefore, a variety of reagents and conditions can cause denaturation. The most common observation in the denaturation process is the precipitation or coagulation of the protein. Let us have a look at the animation to study the effect of physical and chemical treatment on proteins.
Structure of proteins. We will now have a look at the effect of chemical and physical treatments on the structure and reactivity of proteins. Ribonucleus has 2 degree and 3 structures which are destroyed by urea and beta mercaptoethanol. There is loss of biological activity. When all urea and beta mercaptoethanol are removed, then there is a renaturation of protein resulting in regain of shape and biological activity.
Today, in this session, we have learned about primary, secondary, tertiary and quaternary structures of proteins. So, now we come to the end of today's topic. Here's a quick test for you to find out how much you have actually absorbed. First question I'm asking you, name few sources of proteins. Answer is, few sources of proteins are beef, poultry, fish, eggs, dairy products, nuts, seeds and legumes. My next question is, what is the function of proteins in our body? Answer is, protein builds up, maintains and replaces the tissues in your body. Next question is, where does the water present in the eggs go after boiling the egg? Answer to this question is, due to denaturation and coagulation, water gets absorbed in the coagulated proteins. Another question is, name the building blocks of protein. Answer is, amino acids are the building blocks of the protein. Next question is, name different structures of protein. Answer to this question is, different structures of proteins are primary, secondary, tertiary and quaternary. Another question is, who determined the primary structure of protein for the first time? Answer to this question is, Frederick Sanger determined the primary structure of protein for the first time in insulin in 1953. My next question is, what type of bonding helps in stabilizing the alpha helix structure? Answer is, hydrogen bonding helps in stabilizing the alpha helix structure. Next question is, what is the primary structure of protein? Answer is, the linear sequence of amino acids in a polypeptide chain represent the primary structure of protein. That you all have enjoyed today's lesson on structure of proteins. Thank you students for the attention you have paid in understanding these concepts. I hope you have enjoyed the lesson as much as I have enjoyed.